Okay, so this is Bio 2.02 Introduction to Macromolecules, Part 2, because I didn't have my laptop plugged in and my laptop died. And so it cut my video. I didn't intend to make this a two-part video, but it is now. So we had talked about carbohydrates, lipids, proteins, and nucleic acids. So anytime you have a construction project, there's usually a guy standing off to the side with a hard hat, and he has a set of blueprints. And the blueprints are the plan for building the entire house from foundation up through putting the last piece of woodwork on. So the function for nucleic acids is they have the blueprint or the plans to build. Build what you say? Anything. Usually proteins. So usually nucleic acids, proteins are being built based on the nucleic acids. But at some point too, they also have the plan to create new cells or repair cells. And where do we get this from? Well, these come from food, and it comes from all food. Doesn't matter if it's plant, doesn't matter if it's animal. Everything has to have nucleic acids to live. So even a virus has nucleic acids, and we're not even really sure if those are alive or not. So the monomers, we have 5 monomers. And like we have with proteins where we have 20, we call these monomers as a group nucleotides. And we can hook these 20 amino acids together any way we want to. But what's interesting is with nucleotides, those have a specific way that they go together. So we have, I don't know if you've done this before, but we have the expression at CG. What this means is that the two nucleotides, one is adenine and one is thymine, and only adenine and thymine can combine together. The nucleotide 3 and 4 is cytosine and guanine, and only cytosine and guanine go together. Well, what's up with this fifth one? Turns out sometimes when we go from one type of nucleotide to the other, thymine gets replaced with something called uracil. So unlike amino acids that can hook together however they want, our 5 nucleotides, adenine, thymine, cytosine, guanine, and uracil, only adenine and thymine can go together, and only cytosine and guanine can hook together. And sometimes we'll swap out thymine with uracil. But if you need to remember that, at CG is a good mnemonic. We're going to talk about why this is significant later and when does uracil get swapped out. But this gives us kind of a good starting foundation, the function, the source, and the monomers for all of these. And keep in mind, this isn't a macromolecule, that's a macromolecule. So let's sum it up. Glucose is our monomer, and it builds this macromolecule, which we're going to learn is called glycogen. Phospholipid is a monomer, and it comes together to build this macromolecule or this macrostructure called a phospholipid membrane. If I had one little protein, it would probably be right here. So that's one amino acid. So we hook together all of these amino acids to build this structure, which is a protein. So this whole thing is a protein. And similarly, one nucleotide is right here. You can see mine, it's color coded. So this one little red is one nucleotide. That's one nucleotide. And we hook these together to form the polymers that are DNA and RNA. So that's our main takeaway. And that's all we have for the introduction. Thanks for watching and have a good day.